Good morning, children. Today we will start with the sixth chapter, that is Molecular Basis of Inheritance. Mendel suggested that there are some factors or genes which help to maintain the phenotypes and genotypes of organisms generation after generation. After years of investigations of various scientists, it came to the realization that DNA, that is deoxyribonucleic acid, is the genetic material in majority of organisms, while RNA acts as a genetic material in some viruses, but has additional roles as well. It functions as adapter, structural, and in some cases as catalytic molecule. In this chapter, we will discuss the structure of DNA, its replication, the transcription process, that is making RNA from DNA, the genetic code determining the sequences of amino acids in proteins, the translation process, that is protein synthesis, and their regulation. In the last section, the genomics and the essentials of human genome sequencing and its consequences will also be discussed. Now, genetic material is that substance which controls the inheritance of traits from one generation to next. There are some properties of genetic material, like it should be able to replicate itself, it should be stable both chemically and structurally. It should provide the scope for slow changes, that is mutation, which are required for evolution. It should be able to express itself in the form of Mendelian characters. According to this criteria, both DNA and RNA have the ability to direct their duplications because of the rule of base pairing and complementarity using specific processes, whereas the other molecules in the living system fail to fulfill first criteria itself. Example is the protein. Now we will start with the DNA. Deoxyribonucleic acid, DNA, acts as a genetic material in most organisms. It is a long polymer of deoxyribonucleotides. The number of nucleotides or base pairs present in the DNA determines its length. This length is also the characteristic of an organism. Example, E. coli, that is Escherichia coli, has DNA of 4.6 into 10 raised to 6 base pairs. Similarly, bacteriophage Psi-174 has the DNA which consists of 5,386 base pairs. If we talk of lambda-phage, it has 48,502 base pairs, and human beings have 3.3 into 10 raised to 9 base pairs. This is haploid number. Now, let us study the structure of a polynucleotide chain, that is DNA. A nucleotide is composed of a nitrogenous base, pentose sugar, and a phosphate group. DNA is made up of, or it is composed of, nitrogenous base, a pentose sugar, and a phosphate group. If we talk of the nitrogenous bases, it is a nitrogen containing organic molecule having similar physical properties of a base. And there are two types of nitrogenous bases, that is purines and pyrimidines. Examples of purines are adenine and guanine, whereas examples of pyrimidines are cytosine and thymine. Out of the pyrimidines, cytosine is common for both DNA and RNA, while thymine is present in DNA and uracil is present in RNA. A nucleoside is formed when a nitrogenous base is linked to a pentose sugar through N-glycosidic linkage. N-glycosidic linkage or glycosidic bond is a type of covalent bond that joins a carbohydrate molecule to another group which may or may not be another carbohydrate. For example, if we talk of adenine, plus ribose sugar then it is called adenosine. Similarly, adenine plus deoxyribose sugar is deoxyadenosine. Guanine plus ribose is called guanosine. Guanine plus deoxyribose is called deoxyguanosine. Cytosine plus ribose sugar is called cytidine. Cytosine plus deoxyribose sugar is called deoxycytidine. Now, if we talk of a pentose sugar, two types of sugars are present, like a ribose sugar ribose in case of RNA and deoxyribose in case of DNA. Now, let me show you the structure of the ribose sugar and the deoxyribose sugar. This is the structure of ribose sugar. Now, the structure of deoxyribose is like this. This is the structure of deoxyribose. So children, as we have learned, nitrogenous base is linked to pentose sugar through N-glycosidic linkage to form a nucleoside, where glycosidic linkage is a type of covalent bond that joins a carbohydrate molecule to another group, which may or may not be another carbohydrate. Phosphate group is attached to the 5' OH of a nucleoside through phosphoester linkage. The bond between carbon atom 5 of pentose sugar and an oxygen anion of the phosphate group is called phosphoester linkage. And as a result, a nucleotide is formed. Two nucleotides are linked through 3' 5' phosphodiester linkage to form a dinucleotide, and in this manner many nucleotides are linked to form polynucleotide. A polynucleotide has a free sugar at its 5' end and a free phosphate at its 3' end. Thus, the sugar and phosphates form the backbone in a polymer chain, and the nitrogenous bases linked to a sugar moiety projects from their backbone. In RNA, there is an additional OH group at second position in the ribose of every nucleotide residue. Now, double helix model of DNA. Watson and Crick worked out for the first correct double helix model of DNA, which explained most of its properties. The salient features of double helix structure of DNA are: DNA is made up of two polynucleotide chains. The backbone is constituted by sugar phosphate, while the nitrogenous bases projects inward. The two chains have antiparallel polarity, that is, when one chain has 3' to 5' polarity, the other has 5' to 3' polarity. The bases in two strands are paired through hydrogen bonds to form base pairs. Adenine is bonded with thymine of the opposite strand with the help of two hydrogen bonds. Guanine is bonded with cytosine of the opposite strand with the help of three hydrogen bonds. So, a purine bonds with a pyrimidine always, thus maintaining a uniform distance between the two strands of the helix. The two polypeptide chains are coiled in a right-handed fashion. Pitch of the helix is 3.4 nanometer or 3.4 into 10 raised to minus 9. Number of base pairs in each turn is 10. Thus, the distance between the base pair in helix is 0.34 nanometer. Now, percentage calculation of bases is done by A plus T is equal to 100 minus G plus C. The plane of one base pair stacks over the other in double helix. This provides the stability to the helical structure in addition to hydrogen bond. The length of DNA in E. coli is 1.36 nanometer, while in humans it is 1.36 millimeter, while in humans it is 2.2 meters. That's all for today. I hope you have understood the topic. Thank you.